Hi, my name is Jörg Mentje, and I'm the scientific director of the Ludwig Boltzmann Institute for Network Medicine here in Vienna and Austria, and I'm very happy to give you a brief crash course on protein-protein interaction networks. I would like to start by introducing how do we go from individual components, proteins, to networks of proteins. How can we read these interactome networks as a map for biological function? How can we apply what we learned from reading this map in the area of rare diseases, before I close with a brief summary and outlook of what's next. So let's jump right in and start with this really historical achievement of the first draft of the human genome that was published some 25 years ago. And if you have some time, I encourage you to look up the history. It's quite thrilling, the race between public and private companies that led to Tony Blair and Bill Clinton announcing this really historical scientific achievement live on TV. The result of that is essentially, for all 23 chromosomes in the human body, a very, very long list of letters that represent the genetic code. So there are 3 billion base pairs, and the long list, or let's say a short section of this long list, may look like this. And what this draft enabled us to do is, for the first time, search systematically for all the genes that are encoded in the human genome that look like this, that are defined by start and stop codons. And from this, we have learned that we have about 20,000 genes. And this text here is a very short example from chromosome 1. And the 3 genes that are indicated here, they actually code for these 3 proteins. And from this, we understand that just knowing the genetic code and the sequence and, well, the genes doesn't actually teach us that much about biological function, because the biological function really lies in the 3-dimensional structure in the actual proteins. And the next, or one next important milestone scientifically, came 20 years later, and this year's Chemistry Nobel Prize was awarded for this. This was AlphaFold, that allows us to predict very accurately from the genome sequence the actual 3-dimensional structure of these molecules. And the collection of the 20,000 proteins that are, that we consist of, essentially looks like this. And if you think about it, 20,000 is a considerable number, but it's not actually all that much. So I looked at, in preparation for this lecture, I looked it up. There are actually more different Lego pieces than there are proteins. So how can we understand the incredible diversity of cellular function of this multi-scale complex system of the human body, and also diseases thereof ultimately, from the relatively small number of individual building blocks? And the secret is, of course, how they are wired together. So here is a beautiful example where these 13 proteins, they are the components of the ATP synthase, and assembled, the ATP synthase forms this complex. So this molecular machine is really quite amazing. So this is the machine that converts mechanical energy into chemical energy, so that synthesizes ATP. And it's amazing that it sits in the mitochondrial membrane in our cell, and each day it pretty much synthesizes the same amount of ATP equivalent to our own body weight. And the way this looks like is very nicely illustrated in this visualization by David Barry and his team. So you see that it is a rotor, and it converts through mechanical energy that is driven by a proton gradient between the outer and the inner part of the mitochondrial membrane into, well, chemical energy that is then stored in this ATP molecule. Quite cool, no? So we can conceptualize the working of this machine, or the composition of it, through this little graph. So this graph indicates which proteins are actually interacting with each other in the three-dimensional arrangement of this molecular machine, so that the collective function of these 13 proteins really emerges from these interactions. And that graph condenses that information. And over the past 2 decades or so, researchers across the world have mapped out systematically not only the interactions among the components of the ATP synthase complex, but really genome-wide, what are all the interactions that are existing in the human genome, in the human proteome. And what this illustration here shows is that, of course, the interactions that are shown here in the middle between the ATP synthase components are only a very small component of all the interactions, all the physical interactions between proteins in the human proteome. So what do we do with this network then? We have we try to, or in summary, the way we think about this, we also call it in the analogy to the genome, the interactome. We think about it as the blueprint for biological function. So in the same way that the genome represents the blueprint for the individual components, the interactome represents the blueprint of biological function. And the core metaphor for understanding that is the one that this interactome network really provides us with a map. And there are 4 ingredients to this metaphor. The first one is that global shape is important. And I'm sure that you have recognized the Manhattan Island by its shape, and it turns out that also networks have a global shape. And the most straightforward way of characterizing the global shape, if you will, of a network is through the distribution of the number of interaction partners. And in a lecture from yesterday, you heard that this distribution has a very characteristic form that is common to many real-world networks, and that has important consequences. That is, that they are scale-free, or they have a very heterogeneous degree distribution. But there are also other peculiar shapes that are important, such as this ring-like lipid co-regulation network, where the global shape actually is related to the process of inflammation and return to homeostasis in, the human immune system. So very generally speaking, what we have learned over the past decades is that the global shape of networks can be related to the processes that they grew over, and in biological networks over evolutionary timescales. The second part of this metaphor as a map is that location is important. So I'm sure that the Starbucks at this red little flag on Times Square, I looked it up, sells a lot more coffee and is a lot more important in some sense than the Starbucks on the blue location in Roosevelt Island. And the same is true, or the same can be also recapitulated, defined in protein-protein interaction networks. So we can define very simply the centrality or the location of a particular protein through the number of interaction partners. So one that is very highly connected, we would say is central, and those that are less connected are at the periphery of these networks. And very generally speaking, we have learned that those proteins that are in the center of these networks are biologically more important. And here is a very almost a historical example that was published almost 25 years ago, which was the first evidence that the connectivity of a protein is indicative of how important it is, in this case, in the model organism yeast, where those proteins that are highly connected tend to be those that are essential, whereas those that are at the periphery of these networks, they tend to be redundant, meaning that a complete loss of function mutation in these proteins would actually have little to no effect on the fitness of the organism. So the third part of this metaphor is that neighborhoods are important. And in Manhattan, arguably Wall Street down here serves a different purpose, is frequented by a different type of people than Williamsburg. So different neighborhoods have different functions and different character, and the same can also be defined in protein-protein interaction networks. And here is an example that very nicely illustrates that these genes, proteins that are located in the same neighborhood, that are densely connected among each other, they typically are those that perform a certain biological function together. So in biological networks, in protein-protein interaction networks, neighborhood, that is close location, close interaction patterns, are related to being involved in a particular biological process, but also being involved in a particular disease. So this is what we would call a disease module. And finally, the fourth part of this analogy is that distance matters, and I think it is no coincidence that the poorest neighborhood in New York, here in Harlem, is maximally removed from Wall Street at the southern tip. And we can also define the same relationship between neighborhoods in biological networks. And here is an example from work that I've done actually here in Boston in collaboration with Laszlo Barabashi and Joe Loscalzo, and where we could show that the distance between modules related to two different diseases is, very generally speaking, indicative of how related these two diseases are, from having shared biological pathways at the cellular level all the way to shared symptoms or elevated comorbidity patterns if they are very strongly overlapping in these molecular networks, compared to two diseases that have only random associations and no higher comorbidity than you'd expect if the two respective disease modules are clearly separated on these molecular networks. All right, so what do we do with these, what I would argue are very fundamental insights into the nature of how connections between individual components actually shape function? And I would like to walk you through some examples in the area of rare diseases. And to just introduce, rare diseases are defined by, well, being rare. And we have, up to now, there are over 6,000 rare diseases that are described with a known genetic basis. And very typically, these are caused by a single genetic mutation. Now, for a given patient, let's say you have a child that you suspect suffers from a genetic disease, the very first question is, what is the diagnosis? So what is the actual causal gene? Then the second question is, what is the consequence of this particular mutation? So how does a change in the letter in your genetic code actually cause your upstream phenotypes that you are suffering from? And of course, finally, what can you do about it? So what can you learn from the molecular mechanism about potential treatment options? And all of these questions can be translated through these four metaphors that we introduced before. So the first is that when we're looking for the most likely candidate that is actually causing a disease in a particular patient, we would be probably we should be looking here at the center of this network, because we have learned that the genes that are more at the periphery are less likely to have an important impact if mutated. So we can use this basic understanding of the structure of the molecular network to refine our prioritization strategies of variant genes. Second, we have learned that neighborhoods and connections between proteins actually convey biological function. So we can study systematically the connections of this one causal gene to its network neighborhood to better understand the molecular mechanisms that are behind this disease. And we have also learned that drugs typically affect a particular neighborhood in these molecular networks, and we're looking for a targeted treatment that actually modulates the precise biological mechanisms of this disease. So you would look for a drug that has the matching profile on these molecular interaction networks. And I would like to show you some more concrete examples of what does that mean really in practice for one specific group of rare diseases, and these are rare inborn errors of immunity. And to give you an overview, there are some 400, more than over 400 rare diseases that have a strong where the immune system is very strongly affected. 80% of them have a known and well-described genetic basis, and collective prevalence would be between 1 in 1,000 and 1 in 5,000. Numbers are a bit unclear. Now, the challenges, as with any rare disease, is either a missing or at the very least a delayed diagnosis. So for a new patient, what is the actual causal gene? Is it one that has been described before, or is it perhaps a novel gene defect that was not yet associated with a particular disease? And that is typically about 50% of patients suffering from a rare genetic disease actually do not get a genetic diagnosis early on. Secondly, many of these diseases remain poorly characterized. So perhaps one has identified what is the actual genetic mutation causing the disease, but we don't quite know what is really the molecular mechanism by which this mutation actually exerts its effect. And finally, the vast majority of patients do actually not receive a targeted treatment that is specifically addressing the actual pathobiology of the disease, but rather treating the symptoms in an unspecific fashion. So a typical network medicine program would start by mapping the known genes on the interactome, and that is to identify the general neighborhood that is perturbed in this particular group of diseases. So in this case, we would go for hit the internet databases or consult with experts, and this results in our case in 186 monogenic diseases that have an important autoimmune or autoinflammatory phenotype. And the first thing that we would do is to map them on the interactome. So what you can see here schematically is we color the different known genes of these known defects. And the first thing that we would characterize is the so-called largest connected component. That is, how many of those genes actually form a cluster that is directly connected to each other. And what a very typical finding is that they are not scattered all over the place in these networks, but that they form a very significantly sized connected component, and one can quantify that as here by comparing it to the component of just randomly selected genes. And this is what this largest connected component finally looks like. The different colors indicate different subgroups of this group of inborn errors of autoimmunity. And the different colors already indicate that some of these subgroups are connected to each other. So these would be diseases of the complement cascade, but that other subgroups are pretty scattered and actually all over the place. So clearly this more, organ or phenotype-based classification of diseases is unsatisfactory, as it does not quite reflect what are really truly the molecular level connections between these different diseases. So the next thing that one would typically do is, starting from this seed cluster of this largest connected component, we want to identify the larger network neighborhood that represents this disease, and that is what we would call a disease module. And that is what we want to do, is we want to identify those linker genes here indicated in blue that actually connect the different still disconnected component outside of this largest connected component. And one can use a variety of network expansion methods. Essentially, there are different ways to extract additional genes in the human interactome that then connect the existing pool of disease gene to one large single connected component. And in this case, about 213 of such linker genes were required or were identified. A typical rough not proof of concept, a plausibility check is to perform some sort of enrichment analysis, whether these additional genes are likely related to the known biology of the disease. And in this case, we found a bunch of viral infections and cancer-associated processes that are also often related to both the onset but also to the progression of these rare inborn errors of immunity. Okay, so the next step after having identified this larger module that collectively carries the individual components, that is the genes, proteins, as well as the interactions between them that collectively represent a particular disease, is to zoom in and see whether that would allow us to actually define pathobiological subtypes. And the way we do that is to annotate all these individual genes and to then use similarity measures in order to identify clusters of these genes that belong together, that have a certain shared similar function, and that thereby represent subgroups of this disease. And the result looks like this. So this is again this one connected disease module that collectively represents autoimmune and autoinflammatory diseases. And the different subclusters reflect what we would call disease, disease endotypes. That is, these are groups of genes and relationships between them that have a particular, more specific molecular mechanism that then converges to an upstream phenotype. And this redefinition of diseases, if you will, compared to a more clinical categories, provides a more well-defined and actually mechanism-based disease definition, which in turn also allows us to connect the definition or the diagnosis of a disease with a treatment recommendation, because it is really ultimately the molecular mechanism of the disease that you want to treat. And in order to show that, what the authors of this paper did is to first start by collecting what are all the different treatments that are actually used in these rare inborn errors of immunity, and which of those are actually targeted. So which would just treat the symptoms in a very generic, unspecific fashion, and which ones are actually directed towards a particular biological mechanism. And there are only a few of them. And we can then map the known protein targets of these treatments to our interactome and inspect whether the targeted treatments also correspond to those endotype clusters that we have identified here. And it turns out they do. So there is a strong enrichment in those drugs that are actually target, that really operate on the molecular mechanisms. They also fall in the very same disease cluster as the patients that they are given to here in our disease module. So what else can we learn from this network picture? And I would like to remind you that, of course, we are a very multi-scale, complex beast, where relevant processes and length scales range from tiny DNA bases to protein complexes, organelles within the cells, tissues. Tissues are organized in organs. Also, organs talk to each other, and we can think of human biology essentially as a network of, as a nested hierarchical network of networks. And if not the biggest challenge in medicine, but also more generally in human biology, is to understand how molecular level events converge to an upstream, or how they transcend these different levels of biological organization and converge to a clinical phenotype. And it, in turn, such an understanding is what ultimately enables us to also derive mechanism-based and targeted treatments, diagnostics, or perhaps further down the road, even prognosis mechanisms that are really based on a causal understanding of a disease. What we can do with networks here is to try and move from a view that is really focused purely on protein-protein interaction networks, to what network scientists would call a multiplex view that also considers other layers where other nodes and other types of connections are also included. And I want to just briefly run through a work that we have also done in my team in this direction, where we try to understand why is it, or how can we quantify the fact that many rare diseases have highly tissue-specific manifestations, whereas others are syndromic. So how can we understand, how can we even predict perhaps the impact of a genomic aberration on the different levels of the biological organization? And for that, we built a multiplex network of dozens of layers that represent different relationships that are relevant to the human biology, from genetic interactions to also co-expression networks, of course, protein interactions, signaling within cells, between cells, but also functional relationships, shared biological processes, all the way to phenotypic similarities in the consequences of genetic mutations. And the first thing that we try to do is to again relate the fact, or to quantify whether genes related to a particular disease actually cluster in any of these different network levels. And here on this side of this map, this carpet represents the analysis, the results of this analysis. So every row corresponds to a rare disease class, and every column corresponds to one of the different networks in this multiplex network. And the majority of these different networks are networks that are tissue-specific. So what a dot in this matrix indicates is, if it is dark and blue, this means that the respective genes really cluster strongly in this particular tissue. And if there is no dot, or if it is light blue, it means that in this particular tissue, the genes actually do not form a strongly connected component. And we find that the extent to which the genes are connected, form a module in a particular biological network, is indicative of how strongly this particular tissue is actually affected. And we can next use this in order to improve the genetic diagnosis of patients, because we can use the information that we get from a particular patient, that is a detailed phenotypic description of which organ, body, tissue systems are affected, perhaps which cell types, what are known biological processes that have been observed to be dysregulated. We can use all of this information and include it into our prioritization scheme that would weigh the different layers of the network according to how strongly they are affected in a given patient, and use that to improve the ranking of candidate genes in a specific patient. And what this plot very briefly summarizes is that using really the specific network layers that are affected in this multiplex system greatly improves the accuracy of the ranking of different candidate genes in our system. Okay, with this, our time is almost up, and I would like to summarize by taking a step back and give you a sense of how have we used protein-protein interaction networks in research, and how they're also being converted into concrete practical application. And the first step is really always data. So network medicine or network science is really a data-driven kind of science that starts by collecting, well, what are all the known interactions between the essential components of life, in this case of proteins. In a second step, we try to identify what are organizing principles, and in this lecture today I have summarized them in these 4 key metaphors: that the overall shape of a network is important and can be quantified and is related to evolutionary processes. The centrality of a protein in these networks is important and related to how important it is. The neighborhoods in these networks can be defined and related to biological processes, but also to diseases, and that distances between neighborhoods are related to how, well, similar or interrelated these two respective diseases are. Now, the next step is to take these basic insights and try to apply them to different kinds of medical challenges. And I gave you some examples of how we can use, let's say, the centrality of a protein to predict whether it is very likely to be involved in a strong genetic disease. We can use the neighborhoods, that is, the connection patterns between proteins, to elucidate the biological mechanisms, or we can use distances between neighborhoods to identify which drugs are providing a really targeted intervention for a disease or not. And finally, over the past couple of years, all of these very basic insights have also been started to be converted into concrete impact across sectors. So in academia, these are really now part of the standard toolbox of, well, biological sciences. In the clinic, these genomic prioritization pipelines are used, and there are also some very successful spin-off companies here in Boston. Cypher Medicine is one, and back in Europe, we are starting to, or my lab is involved in a very, very large effort to build a European infrastructure for drug repurposing. And I want to close this lecture on a note on how we try to go beyond that. And I would like to show you a little video that of Julia Guthrie. She was the main author of much of the work in the area of rare diseases. And what you're about to see is a demonstration of a virtual reality platform that we have been building, where we include VR for visualizing these very large-scale networks, machine learning and network theory for analyzing it, and latest large language models to really also engage with these data in a very natural way. So here is how Julia would use this platform to diagnose a particular patient, starting by loading up the data and visualizing what are the genomic variants that have been found in that patient. Then next, visualizing what are known other genes related to similar phenotype in the next outer shell. Then inspecting what are the connections between those genes and how can they be interpreted as different biological processes that are affected. How can we use algorithms such as the one that I briefly mentioned in order to rank these genes, and finally invoke some ChatGPT style tools to generate hypotheses on how a particular genetic mutation could actually be causing that disease. And that's it. If you are here in Boston, I would love to see you within the next days, and you can check out some of these yourself. I brought a headset with me. If you are in Rome, well, I'd love to visit you soon, or you visit us in Vienna. You're all very welcome. I hope you enjoy this lecture and the others, and I look forward to meeting many of you. Thank you very much. Ciao.